Okay, so we're going to use the NCBI website. So if you guys want to, you can turn off your cameras and you don't have to have the Zoom up front if you want to switch tabs back and forth, so that I'm going to walk you through it and there's going to be some screenshots, but then I want you guys to be logging in to this site, following along, and just, you know, making up some gene name or just doing the ones that we're doing on here, just to make sure that you can recapitulate these steps on your own with whatever gene that you end up choosing. So, you know, you don't have to go quick look and find a gene. You can just do the fake ones that we're talking about right now, but this will be your job to do that, to pick a gene and look through this stuff, decide if you want to keep it, you know, either keep it or try another gene before next week. And again, if you have issues and you can't do it on your own, then we'll try to do some stuff together next week. But we'll try to, you know, try to keep moving in that direction. So anyway, so NCBI is sponsored by NIH. So you can see that right here. You could get tons of research papers. If you go through the CSUN library to click through NCBI and through PubMed. So PubMed is one of the biomedically related research publication. I want to say suppositories, depositories. Oh boy. And then it also has like where the human genome sequences and all of the model organism genome sequences, programs to actually look at sequences and do analysis. So it's sort of a one-stop shopping. So we're going to go through that guy and show you how to use it. Maybe. Okay. So if you Google, you should be able to find this. So if you search for NCBI on Google, you'll find some sort of page. Now, the other thing is all of these websites change what they look like monthly. So every time I go into some of these resources, like my old list of how you find what doesn't work anymore, and I got to refigure it out. So that's another reason why I wanted you guys to follow along and actually do it, because then you can help me point out where it no longer looks like this screenshot that was made. And so these were made several years ago for our other research class. All right, I'm having issues here. Ooh. Okay. Does everybody find this site? You can shout or, yeah, give me a thumbs up.
Yes.
I can, oh, maybe I can see more of us. Oh my God, it's a teeny tiny little grid now. It still won't show me everybody. Why won't it? Just makes you guys bigger. It still shows me. Okay, I can kind of see a few of you. Okay. Okay. Okay, so if you're at the NCBI homepage, this is the list of like, just, you know, every possible thing. Oh, why is that going crazy? Over on this side, this database is where we would type in to find whatever we're looking to find. This is also where, before you publish something, you're supposed to submit any data or sequences into the databases. And you can download stuff. There's all kinds of tutorials. There's, like we said, tools for data analysis, all sorts of stuff on here. So in this case, it says all databases. That drop-down menu, you can pick specific databases depending on what you actually are trying to do that day. And so here's that drop-down menu. I know it's really hard to see on my screen. So if you guys can click that drop-down menu, you'll see, I don't know, 20, 25 different databases. And the ones that we're interested in right now to look up our gene would be, surprise, gene. And I can't even read what the other one that is actually boxed down there. So yeah, I can't see it even with my glasses. Oh, is that, the hell does that say? Protein? Hmm? Hmm. I don't know. All right, so we're gonna do gene. So before, like it for your real deal, right? You're gonna choose a gene from one of those two lists. So whichever you want, and it might be a little trial and error because we don't really know anything, right? And so an example would be if we pick this guy, and again is expressed not at a higher level, it is on in MCL and off in SLL. We want to make sure that not studied, not characterized, no publications. So the other thing that makes research difficult is every gene has a gazillion aliases, because when something is cloned in a laboratory, and nothing's published on it or that sequence yet, but it doesn't mean you're the only lab working on it. You will spend 2-3 years studying it, figure out what it does, give it a name, and you publish your paper, you upload the sequence. So if all of that happens within a couple months of each other, then there's going to be multiple publications about the same gene with multiple names, because when you were working on the gene, it wasn't published, it wasn't in the database, and then everybody uploads at the same time. Now it's much easier. In the good old days, you had to like mail a piece of paper with sequence on it and they typed it in. So it could take months and months and months for that sequence to be in the database. And publications were out for over a year before they were published. So all kinds of stuff happened in the meantime. Now it's not so bad, but lots of genes have at least 3 or 4 different aliases. And so you can look for those also in this NCBI also gives you lists of alias. The other place that is a good place to look for a list of aliases is in GeneCard. So if you Google GeneCard and then put in your sequence, it will give you, RBP. And then it will list a bunch of aliases, so including all the other ones. And if you put in RBPP8 and that's not what GeneCard named its main name, it'll tell you like the main, oh, no, GeneCard thinks the main name is that, and then it lists RBPP8 as an alias. So GeneCard's a really good thing to look up to find all of your aliases. Because when you're looking for publications, it doesn't cross-reference. So just because this guy that named it RIM published, it's not cross-referenced to then if you type in RIM for it to find CTIP and RBBP8. You have to actually look at all of them. And so that has historically screwed over some students because they didn't do that and they didn't get all the aliases, and then it was published under another name, and so after 2 years of working on a project, they can't publish it because it's exactly the same as what is already known. So that's super important to make sure you get all the aliases. So again, this is significantly a lot of computer work already to go through this stuff, right? So we want to check all the aliases, and then we want to do our searches to make sure, right, it's not out there as a promoter, but we do want to also know what's out there about that gene. It's okay if they study the protein, what the protein does, so the function, because we're not going to do function of a protein or a gene. We're not going to see if it's an oncogene. We're not going to see if it's a tumor suppressor, transmembrane, any of that stuff. We want to know how it's regulated. We are going to study its promoter. How is it turned on and off? Okay, so not that we're not interested in the protein, we want to know the background about it so we know why we should give a crap about why this thing is turned on and off. But we're not studying the protein. So if they know a whole lot about it, that's fine, as long as that knowing a whole lot about it didn't make somebody go, ooh, I want to look at the promoter then, because I need to see when it's on and off in development and blah, blah, blah. So that's why we just need to be careful about function. So it's good to know about its function, and then to make sure nobody's published the actual promoter. Let's see, I think. Takes a while to change slides. There we go. So we're going to look for the gene. We're going to look for aliases in NCBI. We're going to look for the aliases in the gene card. And then once we have all those, we're going to go back to our NCBI. We're going to use that pull down menu. We're going to pick PubMed. I think that must have been that other one at the P down there that I couldn't read. And we're going to type in our gene name with the word promoter first, just because why look up about the protein and what it does and get all hyped up about it if the promoter's already published. And so then we're just going to make sure, right, that this one is about the promoter and not just the gene, because again, search engines aren't perfect. If you put these each in quotes like this into the search menu, right, it'll search for these things separately. So it should still order them by relevance, but you're better off if you're going to do a search and you want to really look for promoter, is put the quotes in and just do your gene promoter end quote. The only problem is somewhere in there it has to have these two things together. If it says somewhere, or the title is the promoter of RBBP8, it's not going to find it if you put these quotes. So you got to play around and do a lot of searching to make sure you're not working on a gene that's already been studied. Because I mean, you'll still learn a lot, but we want to publish at the end. Like that's science, right? Okay.
When you type that, nothing comes up on the search.
Nothing comes up?
No.
Yeah, so try, that's funny. So try searching CTIP promoter.
It's you keep scrolling, it's number 11.
Oh, okay. So this is old, right? If that was number 1 a bunch of years ago.
Yeah, it's number 11. Yeah.
Yeah, and right. So if you're doing live, so it's probably definitely not going to look like what I hope somebody's been studying it. And again, something that didn't have a promoter, like this is this was set up to show like one gene that didn't have a publication versus another gene that did have a publication, or something like that. And then both of them could have a publication by now. So as long as you're finding something, and again, this is about making sure these programs work for you. And these slides will be posted, and then I did remember to start the recording for this part so that you can always listen to me babbling again. So when you do pick a gene and you do the gene card and you make sure you got all your aliases, that when you're looking, if you're like, okay, I'm stuck, I can't remember what the stupid pull down menu, because there's 20,000 things in it, you can go back to the slides or go back to the recording too. And again, this is a research class and we don't do research in a bubble by ourselves. So you're always welcome to collaborate with any of the other students. I'm going to try to set up groups in the groups function of Canvas, and then see if we can make a discussion board for each group so that you guys can talk to each other. I mean, I want you guys to each do your own thing, because it doesn't do you any good if you're the follower and you're just doing exactly what somebody else is doing without any thinking. Again, that's being a technician, which isn't a bad thing. You can make a good living being a technician with somebody telling you what to do forever, but you'd rather be able to think on your own. And so you'll get much more out of it if you do everything yourself and make your mistakes and then figure it out, but you can always get help figuring stuff out. Like you can't remember how to do something and you don't want to listen to this again. You know, and you're like scanning through the slides, you're like, I don't remember it. You just shoot a quick note or post to the discussion, hey guys, where was I supposed to click? Or something we find is totally different than what my slides say. And you're like, oh my gosh, I can't remember how to find that thing. Just ask somebody. It's totally fair game. Right? Research is supposed to be collaborative. You're not supposed to reinvent the wheel for everything. So whatever came up, if you click on that deal, Meyer, if you click on the link, so like, woo, I clicked. Hello, right, there it is. And you can read, the abstracts are all there. Some of these aren't gonna be free. So if CSUN doesn't have a paid subscription to some of these journals, then we don't have access to anything but the abstract. But there's enough stuff free out there in open access and things that CSUN does allow us to have that you can get a good idea about any of these genes. In the real world, if there's something like, it definitely talks about a promoter and we can't see anything but the abstract and it's unclear what they really did, it didn't spell it out. We're not sure if, you know, they were doing like a genome-wide sequence assessment of a promoter, which generally we don't, it's fine if something like that happens. That's not looking at the actual sequences of our promoter and characterizing what proteins bind and don't and activate or repress or so forth. And so if you can't, if that's not free, don't pay $30 or whatever. Send me the link and I just send it to my friends at UCLA who have access to everything and they just download the PDF and send it back to me. So don't pay for things. Absolutely not. CSUN is all about doing everything without having to pay for anything if we don't have to. We know how to bargain shop. And so sometimes just from the abstract, you can tell what this is. Like this one, for example, it says, you know, an intro tells you this is DNA repair and it has a role in gene regulation. You're like, oh, wait a minute, you know, transcription, uh-oh, that sounds like a promoter. But then when you keep reading, its protein is a transcription factor that binds to another promoter. So yeah, it's got, it is involved in transcription, which is, right, that's turning on a gene, transcribing RNA, so might be gene regulation, but the protein of CTIP actually binds to another promoter and turns that promoter on or off or whatever. So this paper is not about the CTIP promoter, it's about how CTIP, the protein, affects somebody else's promoter. And then sometimes even if it's not free, you can see some figures, but sometimes you can't. So again, if it's something like, oh my God, I think this is, you know, the thing that makes me choose yes or no, but you can't open to see the whole thing, then I can get that for you. Are we good? Are we in a page? Does it make sense? Give me a thumbs up or a yell yes. Okay, I'm talking to nobody here. That is the stupid thing about Zoom. It's like, I feel like somebody talk to me. Okay, let's stop weeping. What do we got next? What's next? Okay, it's loading. Okay, yeah, let me see this a little bit. Okay, so now we're back in Gene, and we can find, this is just Gene, right? Yeah. And so apparently in the paper, so this is the reference to the publication, it was called RIM, but I guess it's real, like what, you know, everybody's calling it now, is RBP8. So I think if you looked in GeneCard, that's the name that would come up, and the rest would be aliases, but you could look at that. And now here we also get that list of aliases that didn't used to exist in this database. So now it does list this. So you can also, you know, use GeneCard or just trust that most of them are probably in there. And then, you know, keep adding these guys into here and see if there's more aliases if you put in RIM besides just RBP8. And then make sure for this, we're going to look at human genes. So because the subtraction was between two human tumors, we want to look at the human gene, at least for this, for now. So if you put that in there in gene, do you get something that looks similar to this? I mean, not necessarily this, you know, the exact.
Yeah, it says COM1, CTIP, RIM, SAE2.
Okay, good. So as long as you can find a page that looks similar to this, again, they are always changing the database. So sometimes, like, you know, the side menus are changed, and so, you know, you're probably in the right place even if it doesn't look exactly the same as this guy. Okay, changing slides. There we go. Okay, so then if we go back to PubMed, we do another search, right, for whatever the other gene. So, so this was our example between RBP8 and CTIP. I'm sure there's extra stuff in CTIP now and promoter than there was then. Did you hear that? My neighbor's dog is howling. He's a husky mix. He's so cute. Okay, and so then this was a scroll down, and then the deal, this is probably way down in yours. What year was this? Okay. Yeah, 2006. Holy crap, that was a long time ago now. So it's way down on your page, but it looks like, oh wait, it activates its own promoter and this other promoter. So again, this is just to give you an idea. You don't have to find this exact one. As long as you know how this system is working, you don't have to find this exact and open it. But then, you know, so if you click on it, it'll open again. If it's, you should get the abstract and maybe some pictures of the figures. You can see, let's see, over here, like this says PMC full text free. It's underneath the red thing, but list here. And I think here is just in its own journal is free. So if you click one of these two and it says free, it'll open and you'll get the entire article. If it's not, then all you get is this. Oh yeah, and it also will say right here, free PMC article if it's through this PubMed deal, where like they have to make it free because all the journals were trying to make it pay, and science is supposed to be free exchange of ideas that we shouldn't have to pay for every nickel and dime. Hello, hello. Okay. And then looking at the figures, when you get the whole article, you'd want to look for anything where they kind of map out what a promoter looks like, right? If you didn't watch the the gene regulation stuff and you didn't look at any of the textbook stuff, remember the promoter sits right upstream of the gene. So, right, it's part of DNA, double-stranded DNA. And so they're showing a map of like, here'd be the gene, here's the promoter, and then they're showing that they did some stuff to the DNA. They mutated it, they cut it up, they tested it, they looked at its activity, you know, they did what binds to it, that sort of thing. That's a dead giveaway that they've studied this promoter. Hello, okay. And so there's the close-up of that guy. And so you can see, oh, they've even said they've looked at some transcription factor binding sites and where they are. And here's the sequence. And here's a nice consensus. And they looked at its expression when they made some mutations, which is exactly what we do, right? If you remember back to the lab overview, it's like we clone it, we sequence it, we test it for expression. If it drives transcription, it's a promoter. And then we cut it up, we mutate it, and we see where the parts are that are important. And then we look to see what transcription factors might bind there. And eventually we look to see, does this transcription factor, does that protein bind the DNA? Bingo, yes, it does. We know how it's characterized and regulated. We get to publish it. So if that's already been done, then we don't want to do that gene, right? You pick something else. Yes, yes. Okay. So again, well-studied genes versus less studied genes. So again, we want to know something about them. So way back when we did this subtraction, a lot of the genes were not even in the database, the sequences. And so we named them unknowns, all, you know, that kind of stuff. But now I think everything, even at the time of publication, almost everything was known from when we actually did the study. But if you do a search of a gene name, so this would just be, we pulled one out of the, I don't know if she pulled it out of the list. Professor Hands made this for me. And so I'm assuming some of you already saw this, sorry if you're being tortured. And this guy, and you can see when you search PubMed for any papers at the time, this one only had 2. Okay, so that's really, we know nothing about that. So that's hard too, because then you don't know anything about the protein, but you're pretty sure it was not a promoter study. So you'd be safe to study the promoter. Whereas this one has, you know, at that time, almost 3,600 references or 3,600 publications. Doesn't mean anybody studied the promoter, but you know, it's way more. Now, if this one is 10,000, don't, you know, there's something in there or somebody's gonna do it. But maybe 2, not so great. You know, maybe 50, I don't know, 100, depending on what's out there. So let's go to, nope, new slide. Okay, so if we're gonna think about, after all that, what the heck are we doing? So what are you gonna do, before next Wednesday? What is next Wednesday, the 8th? I don't know. I'm not even gonna guess the date. So you want to pick a few genes to do the test, right? So you can just, you can do it by name. You can, the list has like its major function that was known at the time. So you can, you know, look for something that you find interesting, and you can just pick it by whatever, just whatever. And then you're gonna do the NCBI, right? You're gonna do the gene search. You're gonna get the aliases. You can also check the GeneCard site if you wanna learn more about the databases, right? So you're gonna get all the aliases of that gene, and then you're gonna do the PubMed search. And you're gonna search, I mean, I'd put in the name of the gene and promoter, and I'd try it with without quotes first, and then with. Right, because you want to get some of the protein stuff, and you're gonna, if your one comes up with if you get 10,000 pubs, you're gonna say no, I'm not studying that, right? So that's easy. If you get less, if there's 100, there's 10, right? Then you're gonna say keep looking, right? Then you're gonna really make sure there's nothing about the promoter published, right? And is the gene at all interesting to you? Like, is it, is there some association with some cancer? Is there some association with a neurological disorder or a developmental disorder or diabetes or something else or obesity or bulimia or whatever? Something that, is somewhat interesting to you. Right? And so that's pretty much going to be our job this week, is just to go through all of this stuff. And if once you decide on a gene, then you're going to want to download some of those references, because eventually you're going to want to write a little bit of an introduction, at least for a poster presentation, right? Because honestly, just from this semester, from what we're going to do, you'll, without any wet lab, because everybody knows, that's issues, you'll be able to present what we've learned at the CSUN in the spring. And if we are allowed to do some pipetting, we may be able to do some of our pipetting, this winter, actually in the laboratory, right? If we get far enough in this project. And so then you will have some wet lab data to put on your poster also. So, think of it that way, like you're investing in doing a presentation. And what do all people for graduate schools, professional schools, getting a job, all of these extras set you apart from everybody else. So there's going to be a sea of people graduating whenever you graduate, right? There's however many 300 biology majors from CSUN, the same amount from every other big CSU, a few less from some of the other CSUs, more from all the UCs, thousands, all looking for jobs at the same time, all trying to get into graduate school at the same time. What sets you apart? Think of it, why you? Right? There's going to be a bunch of people with the same GPA as you, or better, right? I mean, not to be rude, but right? There's a bunch of people that took the same classes, regular lecture classes. Now, we have a huge advantage over UCs, just, UC, oh, I can't say what I was going to say. No. Labs. Ever. So not just COVID, they don't have hands-on labs. They have one lab, micro. They have nothing else. They have no upper division labs. They have lots of conceptual. They do lots of like research-based coursework. Nobody knows how to pipette. Nobody, almost nobody. What else do they have? I'm going to say no. I'm going to say almost no. I shouldn't say no. No undergrad research ever. They definitely don't have any this semester. That's what totally screwed over my grant program. But under normal circumstances, they don't have undergrads in their labs. Some labs only have postdocs. They don't even take graduate students. They don't even take PhD students. No master's students. And maybe one undergrad that washes dishes. And if that person washing dishes happens to be super good and smart and everybody in the lab likes them, they might let them help like a technician, just be told what to do, do PCR all day long. You're not doing anything else. And then very few, even fewer than that, actually get to do a real project. So the fact that you guys are even doing this virtual research project puts you at huge advantage over all of this. And so you want to really showcase it, not just that you have directed research on your transcript, but that you've done a presentation. That's huge. That's all science is presenting. Nobody tells us that when we're students, but what do scientists do? Yeah, they do a shitload of experiments, but how do they tell anybody about it? They write constantly. I took English 101 in college, you know, my professor sucked. You don't get writing classes in PhD. You just are expected to write. So you kind of have to learn on your own, and then it's trial and error, and you have to publish and so forth. And you present. You present in lab meetings, you present at conferences, you present everywhere. So, you know, if you didn't take a speech class, like I said, I took speech in high school. My undergrad didn't have very many presentations until I got into a research lab. And I only got into a research lab because it was through the Nerdy Honors Program. And that's one of the reasons I joined the stupid Nerdy Honors Program, is so I could get undergraduate research, because no other undergrads got research. Two of us from my undergrad, and that was a state school. It was Illinois State. It wasn't a fancy University of Illinois baloney. 2 out of the 350 in my graduating class in biology did research. Like, it's huge. Okay, so writing is super important. So you're gonna wanna practice that. And we're gonna practice baby writing, I like to call, which is just for posters, right? The poster intro, posters are supposed to be mostly visual. So we're gonna write, you know, 250 word, 300 word intro. That's nothing, that's like 4 sentences. You're gonna write 1 paragraph, intro sentence, 3 body sentences, concluding sentence, right? And then a little bit of results, a little bit of materials and methods, and a little bit of future aims, a little bit of discussion if we get some results, if we, you know, get to pipette. And we're also gonna do some bioinformatics, so that'll give us results, because that's only on the computer, so we don't have to be pipetting. And then presentations are super important. That's what we do as PhD students. That's what you do for your job. That's what we do as scientists. So the fact that you can say you presented at a conference, even though it's an undergrad conference, that is still gonna kick all these people from UC's butts. None of them ever get to do that. The UCs don't have undergraduate research conferences because there's nobody there doing undergraduate research. So we have them ourselves here at CSUN. And so that's still gonna set you apart at a job, professional school, graduate school, all that stuff. That makes you the one that they wanna hire. Sets you apart from the sea of people that all look the same. And so for presentations so it's CSUNposium, if you haven't heard of it. And it can be as a poster where you give little presentations to individuals, like informally, that come up and ask you questions, or you can do an oral presentation, which is like a 10-minute talk. And sometimes, even though that sounds scary, it's easier to do a talk than a poster, because the talk literally takes 10 minutes and you have to stand and answer questions and do your presentation a bunch of times. That's at least 10 to 15 minutes at your poster for like 2 hours. So you just want to get it over with. And the oral presentation does look better on paper, as far as like, I presented a talk somewhere, right? Okay. So that's why this is so important. And that's why faculty at CSUN are so adamant about getting students, undergraduates, research, even though it takes us tons of time and effort, and all the other students who are working in other labs. So that's why this is crazy too. The 495 has everybody, you know, in one section, even though some people are doing real hands-on work, and then you guys are a virtual lab, and then I have another set of virtual lab people. I'm the only one getting teaching credit for this 495. If the students that are working for Dr. DeBallard or Dr. Flores. Oh my God, now somebody's got a weed whacker. Dr. Flores and Dr. Dablard are not getting teaching credit for spending 10 hours a week with their students in their labs. They're doing that altruistically out of the goodness of their heart. And a lot of times undergrad research is great for the student, but the faculty member doesn't necessarily get a publication out of every single undergraduate. If you can get a bunch of undergrads to work together to get a publication, that's awesome. A lot of times they don't spend enough time and we can't get to that point. And so really it's just because we want to help students get jobs and have careers and get into professional schools and go to graduate school. And so it's really important for the students to step up and do their part to do the research, because it's really all about you guys. So whatever you put into this project is what you're going to get out. And that's not going to affect your grade unless you just stop showing up, right? If you do the things you need to do and come to the Zooms, you're going to get an A. But if you really put your heart into it and really become a scientist, you're going to get so much more. Like it's a huge life-changing event. So with that, I've already talked for way too long. I'm going to stop our, if I know how, I'm going to stop our record. Stop recording.